Hi, Eric.
Hi, Aaron.
Hey, season 4.
Yeah, welcome back.
I'm excited. What are we doing for this season?
Well, I don't know, but it better be good. As you
Huh.
may know, four is a very unlucky number
Oh, it
in
is?
many Asian countries.
Oh, no, you know what? I did know that.
Yeah,
Kind of ghosty or whatever. Well, I'm glad to be back. I had a good summer. And I'm excited to do a new season. This time we're not going to do a book for this year like we did last year.
that's right.
We thought we'd just do random stuff that we thought was interesting and fun.
We have our limits
Yeah, that's
when
right.
it comes to literacy.
Okay, so we're gonna just start. For this episode, there's only one thing that really anybody wants to talk about over the last 18 months.
Oh, actually, I feel like there's been a lot of things, but one of them has been constant.
Yeah, that's right. I remember about a month into lockdown, we had a group meeting at work, and my boss's boss was there, and he was talking about like his interactions with management and their plans for the shutdown and maybe speculation. And we'd been talking about this for like half hour, 45 minutes, and we came to like a stop, and we realized we had other stuff to, like work related to talk about. There was a brief pause, and I was like, guys, this is all anybody wants to talk about, man. We're just so worried. We don't know how long this is going to last.
Yeah,
But today we're going to talk about miracles.
we're pro miracle on this show.
That's right. I thought it'd be fun to start with the Pony Express rabbit's nose story from state conference.
Oh, so I missed that story because while the missionaries were singing, I ran to the bathroom.
Well, our beloved stake president talked about some of his ancestors, and one of which was a Pony Express fella, right? And this guy, so this was during that brief 2-year period where the railroads hadn't been finished yet, but there were still plenty of people out that have moved to the West that needed mail. And so they rode everything by horses, and they would do crazy things like ride for hundreds and hundreds of miles, changing horses in like a 24-hour stretch or something like this. Yeah. And this ancestor of our president kind of his horse ran out of gas, and they stopped in the cold, and they were kind of used, and hypothermia started to settle in, right? And he had fallen asleep, and that would have been it for him usually, except that a little rabbit jumped up on him and licked his nose. Yep, woke him right up, and he got back going and was able to make it on into town. And that little rabbit licking his nose saved his life.
Sounds like it.
And so the point that the president was trying to make, which I thought was great, is that not all prayers are answered directly. Some of them are answered indirectly by like other people and stuff like that,
or rabbits,
or scientists, in the case of our main topic today, which is mRNA vaccines.
Yeah,
which are a miracle. I just want to state that up front. And I think a lot of the scientists who worked on them might be a bit irritated by that characterization. Not all of them are religious, but I think it is.
I'm nearing the end of a monster article in a January issue of the New Yorker that's like, I don't know, 40 pages long or something. Mm-hmm. And it talks about some of the people who literally spent their entire lives working on this, and how the tech just started to come together just when it needed to.
It's absolutely amazing. People worked on these things for decades, and they only came together. They finally started to be clinically useful like a year and a half ago. Just exactly when they were needed.
They were just going into trials for was it a Zika vaccine?
No, it was influenza.
Oh, it was the flu. Oh, okay.
Yeah, from what
I thought it was something more exotic than that. But yeah, they were just starting to work on a potential vaccine for something else when coronavirus showed up. And by coronavirus, I mean a specific coronavirus, but you knew that.
Indeed. SARS-CoV, whatever it is.
SARS-CoV-2.
And then, okay, so what we thought we'd do today is kind of split up the conversation into two parts. One, talking about the history of vaccines in our church, and the other just talk. I wanted to take the opportunity to just tell people about the science, about how mRNA vaccines work and why they're so amazing, and the staggering implications that for humanity in the future.
Yeah, and for all the upsetting news of the last few years, for instance, right now trending on Twitter is the fact that Florida any day now is about to pass, in terms of like how many people have died in COVID, the number is about to become larger than the number of Americans who died in the Vietnam War. So there's plenty of like unpleasant ways to look at this. But a theme of our show going back to the beginning is that we believe that the world is getting better. And I don't think it's unfair to characterize both of our opinions, but certainly my opinion is that this is, shall we say, divinely intentional. Like, I think the world is supposed to get better, and for all the ways the world has demonstrably had a rough year and a half, there's positive things that are going to carry us forward.
And getting better through hard work and sacrifice, right?
Yeah.
And by people being put in the right places and just doing the heavy lifting.
Right. The miracle for 6 billion people on this planet is the hard work of a few hundred or a few thousand that has made that miracle possible.
Yeah. And it's so cool. I can't wait to talk about it. Where do you want to start?
I want to start with the Buddha.
Okay, excellent.
So the Buddha said, and I'm not claiming to quote him directly because I don't speak the language, but essentially something the Buddha said was, if prayer worked the way most people think prayer works, then everyone would be healthy, beautiful, wealthy, and wise. Like if just praying for things got you what you wanted, then that'd be a pretty nice set of miracles, but it would be, like what we would, everything would be perfect. And what it's obviously that is not how prayer works, because if prayer did work that way, everyone would pray and get everything they wanted and everything would be great. So your opening about the rabbit, like things are done indirectly, I think is a really important point we need to keep in mind. The Buddha was right. Prayer doesn't work in this sort of simplistic way that a faithful child might imagine. And it's good to be childlike, but it's also good to recognize that for prayers to be answered, people have to do stuff.
Okay, excellent. That's good. I agree completely.
As long as we're talking about people from other religions, I could talk about Cotton Mather next, if you like.
Okay, sure.
So Cotton Mather was a pastor in Boston. I first heard of him in my junior English class in high school. He was the person we read after Jonathan Edwards. And I don't remember anything about him except his name, and that he was like Jonathan Edwards, sort of a terrifying, but from a terrifying branch of Christianity where God is looking to destroy us. That's all I really remember about what we learned from him.
Excellent.
But so Cotton Mather had a slave. So although I want to point out real quick that although the story I'm telling is available in numerous sources, I like the way it's told by this fellow Michael Harriot, who is he's a writer at The Root, and I'm taking this from his Twitter feed, and we'll put the link in the show notes. Hey Aaron, hey.
Yes.
People want to see this show notes. How could they find them?
Well, if you're listening to the show on your phone, they're just right there. When you go and you look at the podcast on your phone, all of them are right there. Or you can go to faceandhat.com. All the show notes are there too.
Yes. All right. faceandhat.com. How lovely. All right. So anyway, so he gets, I don't know how literally to take this, but he gets a gift bag. And inside the gift bag is an enslaved black man named, I believe it's pronounced Onesimus. This is a name a fellow, for someone who is, living not his best life, enslaved as he is, he's a name that pops up in American history every once in a while. And Cotton Mather did not like this guy. And so just because he was offered to the church as an offering, if you will, didn't turn Cotton Mather into some great charitable soul who loved everyone.
I'm confused.
Anyway. Oh, am I confusing you?
A little bit.
Onesimus was like, he was given to the church as like someone's tithing.
Oh, that's weird.
Yeah, and Cotton Mather didn't like the dude. So because he was the pastor, he now belonged to Cotton Mather in a legal sense, but it didn't make Cotton Mather care for the guy. He didn't really like him.
Cotton Mather was not LDS, or was LDS?
Oh no, this is like early 1700s.
Oh,
like
okay,
approximately
that makes more sense.
1700 exactly. Yes, this is not a Latter-day Saint fellow.
Got it.
So Cotton Mather runs this church in Boston, and Onesimus was not. Oh, Onesimus, I think that's how you say it. He was not impressed by Cotton Mather trying to convince him that God was white. He wasn't impressed when Cotton Mather told him that he was wicked because he was too smart. Onesimus just didn't buy a lot of the racism that Cotton Mather felt he should accept. And this is something I didn't know. And so we'll talk about a little bit about the history of vaccines and inoculations and so forth. Smallpox is kind of the first one that we know of. There's evidence in China of it going back well over 1,000 years ago. Onesimus learned about how to prevent smallpox using these methods from his childhood in Africa. It's not clear how long that was a known process in Africa. It doesn't get to England and Europe until not long before Cotton Mather. Cotton Mather never heard of this. And so what Onesimus told Cotton Mather is that in Africa, people take pus from the sores you get from smallpox and cut you and put it in, and then you don't die of it. And that sounded crazy, but Cotton Mather, even though he didn't believe it, he told the pharmacist friends about it, and they decided, well, why don't we try it? So using pus, they inoculated 248 friends and family that they knew. And as you might imagine, given the nature of humans, as we will see time and time again in history, people went insane when they heard this. They're like, oh my gosh, Onesimus is part of a plot to kill white people. Naturally, and that is America's first anti-vax movement over 300 years ago. So that's pretty exciting. Some of them even they firebombed Cotton Mather's house. So it was pretty serious. Then a few years later, I think it's 15 years, but I'm not entirely clear on the timeline. Almost 6,000 people, which is about half of Boston, caught smallpox. 1 out of 7 people died. I'm not sure if that's 1 out of 7 of the total population or 1 out of 7 of the ones who got smallpox, but whether it's 1 out of 7 of the total population or 1 out of 14 of the total population, that is a lot. But of the 248 people who've been inoculated, 6 of them died. So only about 1 in 40, which is much better. And that Massachusetts then became the first part of white America, if we can call it that, to distinguish it from the native tribes that were here before them, but of British or French or Spanish, whoever. It was the first inoculation program that Massachusetts instituted, which made and it immediately started to work. Another smallpox epidemic hit. Less than 3% of the population caught smallpox, which, if you know anything about smallpox, you know that is pretty great. Unfortunately, one of the anti-vaxxers was his younger brother, who was helping him in the publication of their anti-vax pamphlets and so forth. He didn't want to get his kids vaccinated because he was embarrassed, because he's publishing this anti-vax stuff. Anyway, one of his kids dies from it, and this gentleman will go on to open the first hospital in America. He'll also go on to start the first fire department in America. He'll open the first public library in America. Do you know who we're talking about, Aaron?
Oh, are you talking about Franklin?
Yes, Ben Franklin.
No way.
And he regrets not or it's not a vaccine technically, it's an inoculation, but anyway, he pushes for mandates. Thomas Jefferson got vaccinated in Philadelphia, and so he wasn't so much worried about it. Hang on, I have a really great phrase, and this is from another article. This is from Nature, actually. So in contrast to Europe, where smallpox was an endemic illness of childhood, in the colonies, smallpox occurred in large epidemics with about 20-year periodicity and affected children as well as older susceptible individuals. This became an important issue during the Revolutionary War, when the troops of the Continental Army, but not the British forces, were ravaged by smallpox, until, tell me if you've heard of this guy before, General George Washington at Valley Forge decided to inoculate all susceptible troops. This act probably saved the war effort.
Wow. This is incredible. I didn't know any of
Yeah.
this.
Yeah, no, I didn't know any of this until fairly recently either. And I could kind of go on. The history of it is fascinating, but the point is that can I tell you one more story?
Oh,
I
yeah.
really like this story. This is not American history. It's not US history, it's Mexican history. But so inoculations and all this had been discovered, and in England, Jenner, is it William Jenner? What's Jenner's first name? Do you know? It doesn't matter. He discovers, like, if you get people sick with cowpox, they won't get smallpox. And I believe it's the cowpox that's in this story. I should be double checking it, but people can look it up. It's not that hard to look up. It's called the internet. But anyways, whether it was smallpox inoculation or cowpox inoculation, the point is that they collected all these orphan boys who needed a better life and said, hey, here's the deal. We're going to get 2 of you sick with cowpox or smallpox or whatever it was. You're going to go on the boat with us. And when you get to the pus stage, we're going to take some of your pus and inoculate 2 more of you. And then when they get sick enough to have pus, we will use their pus to inoculate the next 2, with the idea that we can cross the entire Atlantic Ocean and get to Mexico and still have, and then we can inoculate the population there. And the crossing took a little bit longer than they anticipated. When they got there, there was only 1 kid who was left, and he was in the pus stage at that point. And from his pus, a nation would be inoculated against smallpox.
This is bananas.
Yeah, it's a crazy story, but it sounds like, and on one hand, it's really unethical. On the other hand, like, these kids got a pretty good trade, really. They got a new life, and they were given money, and I mean, it would not work today.
Yeah, probably.
Yeah, but when you consider the possibilities, the options they had then, it was pretty good solution, really. I mean, it's a little icky, but I can't come up with a better solution given
it's pretty
tools
icky.
they had at the time. Yeah, no, it's terrible. But given the tools they had at the time, it's you could really make a utilitarian argument. It was the greatest good for the greatest number of people.
Wow.
Yeah. So I haven't got to the church yet. As far as, I couldn't find anything. I looked through a lot of sources. I couldn't find anything about inoculation in church sources until the mid 20th century. But from the moment it shows up, it's treated as common sense and valuable and important.
Hey, folks, jumping in from the future. The first presidency recently helped us out here by sharing a pro-vaccination letter from the 1900s. It's a great little look into some history, and we've put it in the show notes.
On the church's humanitarian page, they proudly proclaim that every year, well, they don't, let me rephrase that, because the first sentence is not a proud proclamation. It says 1.7 million children die from vaccine preventable diseases every year. And then the proud proclamation: the church provides support to international immunization campaigns for measles, diarrhea, and pneumonia. Between 2003 and 2012, the church supported campaigns that benefited over 100 million children and youth in 36 countries. Around 59,000 local volunteers donated over 750,000 hours to spread awareness of these campaigns. And it's kind of difficult to get very far through the humanitarian services without finding something about vaccines. For instance, Latter-day Saint Charities, the humanitarian arm of the Church of Jesus Christ of Latter-day Saints, is giving $20 million to UNICEF to help distribute 2 billion COVID-19 vaccines to at-risk populations around the world. It's already reached 121 countries and territories. Obviously, if you follow the news internationally, like, the vaccine is not everywhere yet. People are still trying to get it, but the church is 100% dedicated to helping distribute what you term a miracle to the world, which I think you could argue is largely what the threefold mission of the church is: to distribute miracles, right? Whether that's salvation or vaccination,
Right.
it's
I mean,
much in the same. All things temporal or spiritual.
especially the more modern reinterpretation of that phrase, right? Where the old phrase, perfect the saints, redeem the dead, proclaim the gospel, right? It kind of got rewritten, I don't know how many years ago, and essentially included care for the poor.
Yeah.
So in this case, that makes a lot of sense. What about, like, did you happen to, in your readings, I was wondering what, like, in the LDS community, like, reactions to things like the polio vaccine and things like that.
As far as polio is a really interesting one, because there's less evidence that there was a real backlash against polio. And I think the difference is with polio, it really only affected children. It was kind of the opposite of early COVID. When COVID-19 first showed up, it didn't seem to affect children. And polio was the opposite. It only seemed to affect children.
Yeah.
And I think that caused a different reaction. I mean, I think if Delta had been the first variant, we might have reacted a lot differently.
Because kids were getting it.
Yeah, because kids are getting it. Kids are dying. I think that I read something today that it's the most it's the most contagious disease known except for measles, the Delta variant. I'm not exactly sure how that's measured. I didn't read the article. I just saw the headline, but I thought it was
it's measured using a number called R.
Yeah, the R naught. I do know that, but I also wonder, like, does that include, say, smallpox? Does it include extinct diseases? I should have read the article, but I didn't know it was going to come up in our conversation.
There's so many sources for today's episode.
It's true.
Awesome. Well,
sorry. I was just going to say, I remember when on our first COVID-19 episode, how you talked about how all the scientists were working on the same problem and how unique and interesting that was. And that gets to your...
I have a reading for you about that.
Okay, let's have it.
Okay, so we're actually going to be doing several Nature articles today. So do you know what the big three are? The big three journals?
Obviously Nature and Science are two of them. I'm going to go with Cell.
Cell is the other one. That's right. So we have this will be the second reference to Nature article today. This one is called The Sprint to Solve Coronavirus Protein Structures and Disarm Them with Drugs. This was published on the 15th of May, 2020. So,
wow, that's so quickly.
Yeah, so by Megan Scudellari. All right. Lying in bed on the night of 10 January, scrolling through news on his smartphone, Andrew Messakar got an alert. He sat up. It was here. The complete genome of a coronavirus causing a cluster of pneumonia-like cases in Wuhan, China had just been posted online. Okay, so the complete genome. So we're going to come back to what that means later. Okay, but what it essentially means, it's the full DNA sequence, or RNA, because viruses are RNA sequence of the coronavirus.
Yes.
Around the world, similar notifications appeared on the devices of scientists who first crossed swords with coronavirus in the 2003 outbreak of SARS, and then again with MERS in 2012. Instantly, the researchers mobilized against a new adversary. We always knew that this was going to come back, said Messakar, head of biochemistry at Purdue University in West Lafayette, Indiana. It's what history has shown us. In Lübeck, Germany, Rolf, and the names here aren't important to remember. It's just fun flavor.
Very international.
In Lübeck, Germany, Rolf Hilgenfeld stopped packing boxes for his retirement and started preparing buffers for crystallography. In Minnesota, Feng Li stayed up all night analyzing the new genome and drafting a manuscript. In Shanghai, China, Hai Toyang rallied a dozen graduate students to clear their schedules. In Texas, Jason McKellen instructed laboratory members to start assembling gene sequences from the viral genome. Within 24 hours, a network of structural biologists around the world had redirected their labs toward a single goal: solving the protein structures of a deadly, rapidly spreading new contagion. To do so, they would need to sift through the 29,811 RNA bases in the virus's genome, seeking out the instructions for each of its estimated 25 to 29 proteins. With those instructions in hand, the scientists could recreate the proteins in the lab, visualize them, and then hopefully identify drug compounds to block them or develop vaccines to incite the immune system against them. Here we go, thought Messakar. I'd better get some sleep. And then it keeps going. The article is fantastic because it goes through the first few months of the scientific effort and how they, and it keeps a clock of how many cases there are as it goes, and the work on the structures of the proteins.
I had no idea. I mean, first of all, it's still amazing to me that how quickly we can sequence nucleic acids.
That's right.
I find that astonishing. And then I didn't know the same information that you just read was also in that New Yorker article I mentioned. I didn't realize that just by looking at the base pairs you crazy scientist people can now determine like what a protein looks like just from reading the base pairs. Like,
Well,
I don't know how
sort
that
of.
works. That's really impressive.
Sort of. We still had to crystallize the proteins and solve their structures. I mean, that's what I do for a living. I'm a protein crystallographer. Let's go ahead and stop for a second and do some high school biology. I want to refresh everybody's memory as to what these things are that we're talking about, right? And I apologize if this is if this is too basic, but I really want to talk about how awesome mRNA vaccines are. And so to do that, we kind of just need a quick refresher, okay? So what are the components of the cell?
Well, you have a membrane.
Right, that's one of them.
You have a nucleus that has its own membrane.
Mm-hmm.
Have...
so I'm not talking about the organization within the cell.
Oh, okay.
I'm just
We're not
talking about
mitochondria, the powerhouse. Well, you're talking about something else.
I'm talking about what are the basic building blocks of a cell.
I'm going to say carbon and
that's more basic than I meant.
Oh, dang it, I'm bad at this.
Proteins.
How about proteins?
Yep.
And we have those nucleic acids we were just talking about.
Yep, membranes is the third.
I suppose there's water, of course. We have
water
and then sugars.
Yes, and so I'm going to call it everything else.
Okay, everything else. That sounds great.
So you have membranes. And membranes are the walls of the cells.
Yes.
Okay. And it divides up the cells into compartments, like you said, like the nucleus and so forth. All right. And then the proteins, they're the machines. And proteins are amazing. Okay. Yeah. There's so many different shapes and structures, and they're built up like Legos. So if you think about a Lego set, you know, how many different Lego pieces are in a Lego set? Maybe you could do a lot with 20 different kinds of Legos, right?
Sure.
A 2x4, a 2x2, a 1x2, right? Everybody has their own names for Legos that they came up with as a kid. But given like 20 different shaped Legos, you can do a lot.
Sure.
And that's what proteins are. They're the machines. They're engines, they're cranks, levers, enzymes that catalyze really difficult reactions. And if you think about the pictures of the coronavirus, the red and white ball with the green spikes on it, right? That's all the outer shell of it, and that's all made up of different proteins that have come together, interlocked, and formed this cohesive unit. Some proteins even walk along like superhighways in the cell. They're really cool. Okay, then comes the nucleic acids that you mentioned, DNA and RNA, right? DNA is the blueprints. All right. They code for the proteins. All right. DNA is transcribed into RNA. RNA is translated into protein. All right. And so those keywords help you understand what's going on. The RNA is a copy of the DNA. And it's sent from the nucleus outside to the rest of the cell, where it's translated into protein. And so that's how that works. And so so that's just the start. That's where we're going to start with, that basic high school biology. Yeah. So with that in hand, we can talk a bit about the immune system, right? Do you know, like maybe just give a quick summary of how the immune system works? I'm really grilling this English teacher here on high school biology.
No, that's okay. Like I tell my students that English class is every class, and every class is English class. And
oh, that's great.
We do a lot of basic science, actually. Some math, some statistics, all kinds of literacy are fair game in my class.
Excellent. Okay, so the immune system, how's it work?
Okay, well, something appears that shouldn't appear.
Yeah.
And there are lots of tools available. You got your T cells, you got your white blood cells. I'm going to try to be simple, but essentially something shows up and the body starts throwing a lot of stuff at that to see what works. And when it finds something that works, it quickly destroys the thing if it can. Of course, there are other stuff happening too. Your body, cooks itself in order to kill bugs. And
it's a multi-pronged attack.
Yeah. And sometimes, and this is something that COVID seems to be good at triggering, it can cause, like, the immune system can end up killing you because it rages out of control as it's trying to destroy the invader. Or
That's right. And so the principal response is to make antibodies, right? So the antibodies are made to the antigens. And so the proteins that coat the coronavirus, those spike proteins and all the other parts of the proteins, those can be considered the antigens, right? And the antibodies are going to be made by the immune system to match them. So it's like this. If the antigen is a key, the antibody is a lock. Only in this case, we have one key, and the body has already made 1 million different locks before the virus even arrives. And it tests them all on the key to find the right one. And they find the one that fits the best, and it doesn't fit quite good. And so they randomize the lock a little bit, and then they keep going until they have found exactly the match, right? And then then the immune system memorizes that forever, or however long.
Hopefully.
Hopefully. And so
it's better at remembering some things than other things, it seems.
Yeah. And then from now on, if it ever sees that antigen again, it can attack it immediately. So that's how the immune system works. Okay, so all of this was really kind of discovered in the early part of the 20th century, right? They all kind of started work, and in mid part of the 20th century, all of this immune system stuff started to get worked out. All right. Now I'm going to jump all the way to 2020, and I'm going to tell you that the Moderna vaccine was ready to test less than a month after the virus genome was published online. All right. And an army of volunteers, hundreds of thousands of people, received the trial as a placebo, and a blind trial, right? So they did the controlled trials. Some got placebo, some didn't, and they were ready to start testing it. So how did we get from a rudimentary understanding of the immune system to less than a month and having a vaccine ready to go? How did that possibly happen? And so there's 3 sources that we're going to use. All right.
Okay.
The first source is an absolutely excellent video, and it's called "Why It Actually Took 50 Years to Make COVID mRNA Vaccines." All right. And it's on the SciShow channel, and it's hosted by a guy named Hank Green. And it's excellent. You could go there and it's a 12-minute video, and you'll just learn a ton about how come it was so fast and yet took forever.
Is that Hank Green, the vlog brother Hank Green?
I think so. He's one of these internet style like personality fellows.
Yeah, one of the first ones. He's pioneer.
Excellent.
Anyway, go ahead.
All right. The second source, it's an article published in UAB News, the University of Alabama at Birmingham. Right. And the article is "COVID-19 mRNA Vaccines. How could anything develop this quickly be safe?" Written by Nam Moon. Moon.
Well, I'll admit, and I am not by my nature anti-vax at all, but I was caught a little off guard by how quickly it happened. I felt some suspicion also. I was like, this is nothing like this has happened. There's no precedent for it. And I was a little skeptical at first for exactly this reason.
Well, you and many people.
Right.
And the third article actually was published the 14th of September 2021. That's 2 days ago as of recording. And it's called The Tangled History of mRNA Vaccines in Nature by Ellie Dolgan. So those are the 3 sources for the science that we're going to cover, and I recommend you look at them. In fact, Eric, I'm going to share my screen for you really quick.
Let's do it.
And I just want to show you this picture from the Nature article right there. And do you see can you describe what you see?
I would love to. So I see what I think are base pairs in the background.
Yes.
And then, you guys should understand, I'm not looking at like an important graph or something. I'm looking at an illustration. But the background is base pairs, vaguely matrixy with, in a computer font, but it's blue instead of green. And in the foreground is a sequence of rectangular shapes, which I realized a moment later were a hypodermic needle.
That's
And
right.
inside the, what do we call it? The chamber? Like, what do you call the,
Sure.
okay. Inside the chamber, there is a circle, which is made up of little ovaloid shapes connected to each other in a squiggly line in the middle, which I'm going to guess is the mRNA.
The
Is
squiggly
what
line
that
is the
represents.
mRNA. That's right. And the
I don't know what,
circle
yeah,
is
what
the
is the circle?
It's called the liposome.
Is that supposed to protect it long
It
enough
is.
to?
It is, and it's absolutely critical. We're going to talk. We're going to talk about that in a minute. What I wanted to show you this picture is because this RNA sequence is the COVID-19 vaccine.
Oh,
Okay.
right, right.
That's it. That's the entire thing. That's the COVID-19 vaccine that you see right there. I mean, how many lines of code is that? It's like maybe 40. 40 A, B, C's, and in this case, the Greek letter psi, which is standing in for use.
Why?
That's going to be very important later.
Okay, then I'll wait. Can I just jump in though and say that, like, a piece of RNA, that is such an improvement over taking pus from someone and scratching you and rubbing it into your body.
It sure is.
So much safer and such a huge improvement. And I'm very enthused about this improvement.
All right. So it took decades, right? So the first thing that we want to talk about is in the 70s. Early experiments in the 1970s discovered that if you take mRNA, the M here stands for messenger, right? So the DNA is transcribed into messenger RNA, mRNA.
Wait, can I ask you a question?
Yeah.
I think I know the answer, but I'd rather get your answer on record rather than my guess.
Yeah.
Why is it? Because as you described RNA to us earlier, its entire purpose is to be messenger. So why designate it from other RNA with the M?
There's two other kinds of RNA called tRNA and mitochondrial RNA. And both of those are used in different ways. RNA is different than DNA by taking, so a DNA is ATCG. That's what the letters are in DNA. You take all the T's and change them to U's. Now you've got RNA, right? And so that RNA, to then how you use it, gives you the little letter in front of it. So mRNA is for messengers, tRNA is used in ribosomes, rRNA is also used in ribosomes. So there's a bunch of different ways to use RNA. And so mRNA, message RNA, this is the RNA that is a gene that's been transcribed, meaning copied from your DNA, and is going to be translated into a protein by a ribosome. Okay, so early experiments tested adding mRNA transcripts to cells. They took rabbit mRNA and they added it to frog cells, and they got back rabbit proteins. So in other words,
so the frog cells were creating rabbit proteins because the rabbit mRNA was stuck in the frog.
That's right. Exactly right. So they realized that this could be very useful as a tool. In that case, they were only interested in using it as to study like genes and how they interact and things like that. It wasn't until later that someone realized that it could be used as a drug. Okay. So in late 1987, a guy by the name of Robert Malone performed a landmark experiment. Here I'm reading from the Nature article. He mixed strands of messenger RNA with droplets of fat to create kind of a molecular stew. And then human cells bathed in this genetic gumbo absorbed the mRNA and began producing proteins from it. Okay.
Sounds like the worst YouTube cooking show ever.
Realizing this discovery might have far-reaching potential in medicine, Malone, who was a graduate student at the time, right, wrote in his journal in 1988, it might be possible to treat RNA as a drug. And so this was the first time anybody had ever used fatty droplets to ease mRNA's passage into a living organism. So what someone realized is that they could make a human cell, change its behavior by giving it mRNA to do something different, right? And once you can do that as a toolkit, you can do so many different and interesting things. But they're found lots and lots of problems. And they had to solve all of these problems by 2020, and that's for us to be able to have the mRNA vaccine as we have it. Okay, so let me tell you what some of the problems were. Okay. Oh, so why the fatty droplets? Let's talk about fat first, because that's kind of weird, right? So the mRNA, if you just imagine you had like a handful of mRNA. First of all, what would it feel like? What would its color be? I don't know. I don't actually know the answer.
It'd probably
be white. And so then you ate it. All right. What would happen?
If you just ate it, yeah, it would break down immediately.
Yeah. So that's not going to work.
Yeah.
Okay. Well, and instead, what if you just okay, so that's not going to work. So let's just inject it directly into the bloodstream. What happens then? The same problem. It just gets degraded. Your cells, they absolutely hate just free floating mRNA, because it thinks that it's a problem. It thinks it's like from a cancer cell or from some other kind of problem, right? And it immediately attacks it and degrades it.
Yeah.
The immune system actually is recruited to degrade it, right?
I suppose it all gets recycled too, all those useful pieces.
In fact, it's so common that one of the most ubiquitous proteins is called RNase, and it breaks down mRNA. That's what it does. So I actually used to work on RNA. I worked when I first moved to the Bay Area, I worked for a scientist named Jennifer Doudna, who you may have heard of before.
Is she the at Cal?
She is at Cal. That's right.
Yeah, she's the CRISPR lady.
She's the CRISPR lady. Now, when I worked there, I didn't work on CRISPR at all. I worked there for a couple of years and then moved on to other projects at Berkeley Labs. I would do so.
That's why you don't have a Nobel Prize.
And there's many other reasons, but she does. Okay. But I think the important, and so Jennifer Doudna is awesome. She's one of my heroes. If you want to just learn about someone interesting, go learn about Jennifer Doudna. But important bit is that it was an RNA lab. And so what I had to actually work with RNA, and before I could even work with it, I would have to spray down my working area with this thing called RNase zap because it was everywhere. The RNA that I would be working with would just degrade if I wasn't incredibly careful. It's all over your fingers. It's all over the walls. RNase has coated our lives, and because cells dislike it so much. So it's unstable. When scientists started trying to use it as a potential vaccine, a lot of funders laughed at them and said, there's no way. It degrades too quickly. You're never going to get it to survive inside a cell. So that was the first problem that they had to solve. And here's how they solved it. Okay. And if someone's going to get a Nobel Prize for our mRNA vaccine system, it's going to be these two people who I'm going to talk about next. They're at the top of the list. All right. So they are Caitlin Carrico and Drew Weissman. They partnered in the 90s to start working on mRNA delivery systems. And they worked on it for quite some time to try to figure out why it was unstable, why the RNA was unstable. And what they found was that when your cells make RNA transcripts, they change the U's in the RNA to pseudo U's. That's what that psi symbol, the trident, the Greek trident was. It's a small little modification to change uracil to pseudo uracil. And the cell now won't attack it. It thinks it's its own RNA and not foreign.
Oh.
These results were published in 2005, and it was a bombshell, right? I mean, from what I understand, it was a big deal because now they could do things with RNA that you couldn't before.
Interesting. It seems to me, I don't know how the taxonomy of intercell molecules works, but it seems like if it's a different molecule, it should no longer RNA. It's something different from RNA.
It's a good question. So RNA is comprised of 4 letters, just like DNA is.
Right.
So DNA, ATCG, RNA, AUCG, right?
Which is why it's not DNA.
That's right. This is pseudouridine. Dear reader, I'm showing a chemical structure. All right. And then this I'm hovering here is uridine. Mm-hmm. And you can hardly tell the difference between them.
I don't. Oh, I see it. Yes, it is very subtle.
It is very subtle. A nitrogen atom has been flipped.
Yeah. I missed it on my first pass.
Yeah. And so when you make your RNA using this, it still acts as RNA and it's fine. The cell can use it, no problem. But the immune system doesn't target it.
Interesting.
Or rather is attracted to it enough that it can start to learn to incorporate the RNA and start to make the antigens.
So get on a time scale, let's say you made the Moderna vaccine using uridine instead of pseudouridine. What would be the time difference in how long it takes the cell to realize it doesn't belong there and destroy it?
Oh, I mean, it doesn't, from what so that's a good question. It kind of leaves what I've studied, right? I don't know how long, what the degradation time is for unmodified RNA versus modified, right? What I do know is that it's even debated. Some scientists think that you don't have to do this, and instead you just have to change how you purify the RNA. You just have to make the RNA better, and then it's not a problem anymore. But other people insist that it's required. It's actually still up for debate.
So we don't know yet.
We don't quite know yet, but the preponderance of the evidence is that using pseudouridine allows it to be targeted correctly. Okay, so that was one problem that had to be solved, but it's not the only one. All right. Okay. They also figured out that they could modify the cap, which is like the head of an RNA transcript. Think of an RNA transcript as like 1/2 of a zipper. That's kind of what it looks like. So they just changed the head and the tail of it, and that also makes it more recognizable by the cell, keeps it from being degraded, right?
Sure.
And that's fine. But now they needed a way to get the RNA into the cell walls, and now this is where we come back to the fatty droplets. Okay. So think about a soap bubble, right? A soap bubble forms spontaneously from soap and water, right? What it's made of is a membrane. Well,
right.
A cell is a membrane as well. It's actually the same thing. How did you make soap back in the day, right? You rendered fat to produce the soap. Soap is fat in some ways. It's not quite right, but that's kind of what it is. Enough. But it forms bubbles. Yeah. The important bit is that it's a bubble, and cells are bubbles. And so you can, if you wrap the mRNA in these bubbles, it now the cell can glom onto it and bring it in. But it's even more than that. It has to be the right composition of the bubbles. What they discovered was if they use these particular fats that have a strong positive charge, that would counter the negative charge of the mRNA, and that would allow the everything to kind of equilibrate better. And then, but also when you inject them and you hit the blood, that kind of falls apart, and it becomes less toxic or not toxic. So anyway, but so you can read more about this in the article. But the point is, there are these innovations that they had to do this, and each one of these innovations came from different scientists. They're all the entire group of people really are eligible for these Nobel Prizes when they come out, in my opinion. And the effort is it's kind of comparable to a moonshot when the scientists in 2020 started running, right? But also you have to think about all the work that came ahead of time to do it. Decades of research to get this all together.
Can I, sorry, this is a little backwards, but can I ask a couple lipid related questions?
Yeah.
One, correct me if I'm wrong, but would it be accurate to say that this issue of how as you were describing, like the soap membrane and so forth, like soap, literally, this is why it kills bacteria and viruses, right? Because it's destroying the lipids on their surfaces. Is that correct, essentially?
I don't want to say yes or no.
Okay.
I think that it's more like there are some detergents that can break up their cells that are essentially kind of tear them up.
And here's my second question. This is more related. Viruses also are surrounded by lipids, right?
Some of them are, not all of them.
Coronaviruses are, though, aren't they?
They might be.
Anyway, okay, maybe you don't have the answer to this one either, but I was going to ask if this is part of the virus's strategy also, is to use the lipids to
coronavirus is an enveloped virus. You're right. It has a lipid layer on it.
Who's the scientist now, Aaron?
Hey, we're all scientists when we do the research. So that's pretty much what I wanted to say about mRNA vaccines. Oh, of course, the last bit is the actual payload of the vaccine, which is the mRNA sequence, which is what we were just looking at there. So that codes for a section of the spike protein, which is one of those little spikes on the COVID particle surface, right? Mm-hmm. And so what happens is the cells absorb the fatty phospholipid thing and the liposome, and the mRNA goes into the cell. It transcribes the spike proteins and then excretes them, and then that promotes the immune response, and then you're good to go.
Yeah.
So okay, so the point is that, isn't it amazing that all the research finished at just the right time?
Yeah. I mean, it's an incredible set of coincidences. It would be really bad screenwriting. It's a total Deus ex machina, and it feels like cheating that we got this so fast. But that's what miracles are, I suppose.
I'm so grateful for them. I've been able to see friends that I haven't been able to see who are all vaccinated. I've been able to be less afraid. It's just awesome. And then, and this is the part that I'm actually kind of the most excited about. People are taking the mRNA vaccine approach, and they're using it to like create like other kinds of vaccines, like flu vaccines, like we talked about before, mm-hmm. but also like cancer treatments as well. And so you can do the same thing. If there is a cancer cell that makes a kind of protein, kind of a deformed protein that normal cells don't make, then mm-hmm. you can create a vaccine against it. And then your immune system will be targeted right to the cancer and take care of it for you. And that's just amazing. I mean, and then there's ideas that people have about doing like treating genetic disorders. And it's just fantastic. I'm so excited by it. There are some things that are going to change the world, and this is one of them. This kind of technology, this kind of medical technology, this is really when we talk about how God wants us to change, make new medicines is one of them, and this is very exciting.
Yeah, that's in Come Follow Me. I guess it was really more last month. We were going through a lot of sections about learning from the best books and learning the nations and the sciences and everything we can about this world of ours. And it's pretty exciting that there's still things to learn, still ways to improve things.
Okay, if we got anything wrong, you guys can please let us know. We'll correct it.
Yeah,
I'm excited for season 4. Next time we're not doing anything science related, and it's going to be fantastic.
Yeah, that does sound fantastic.
And
no, I like the science. Hey, Aaron.
Yeah.
So as you may have heard, what with us being a proud member of the Dialogue Podcast Network, but there are some new people joining the network, including my friend Madison, his podcast Bristlecone Firesides, which is about the intersection of being a Latter-day Saint and ecological and environmental questions. And Blair Hodges is doing the Fireside, which exciting new interview show. So there's lot of exciting things happening right now, in addition to our fancy new theme song. Thanks to
oh, right here. Thanks to Daniel Foster Smith.
Yeah.
We're very excited to be kind of leveling up the show. So thanks to him. If you want to find us, you can find me at Aaron Brewster on Twitter, and Eric is
THMAZING.
Also, you can find the show at Face and Hat on Twitter, and you can find it at faceandhat.com. We have a Discord server now for the show. I'm going to put the link to it in the show notes. You can jump in and chat with us there. Do you use Discord for any other purpose?
I do. The Association for Mormon Letters has a Discord, and I joined a Discord run by Mormon artist Lazaros.
All right. Yeah, you can find me
better than Facebook
there. Oh, yeah, for sure. All right. That's it. Bye, Eric.
Bye.